Good day, guys. You guys are welcome to this tutorial. We are still answering our past questions on physiology. And this question, and the students in an examination were asked to write on the regulation of genetic activities. Okay, students in this examination were asked to write on the regulation of genetic activities. All right. So I think let's attempt answering this question in our own way. All right. So genetic activities, regulation of genetic activities. So now we say that first, the genetic activity of a cell is the genetic activity of the cell is to produce proteins. Okay, um, the control of protein synthesis is based on factors such as use is the protein needful, all right, and available or availability of the resources. Okay, and in availability of the resources, you have something like okay, all right, this protein is useful, but do we have the resources to actually create this protein that we are talking about? Now, first of all, is the protein useful or needful? And secondly, the availability of the resources. Do we have the resources to produce this protein? All right, that's the genetic activity that we are talking about. It just has to do with the production of proteins. Okay, now this is an operon that we are talking about. You see the regulatory gene here, you see the promoter, you see the operator. All right, we get to talk about all this. All right, so first of all, we say that the chief regulator of protein synthesis is what? Operon. This is operon that we see here. Right, now operon are set of what? Nucleotide that are involved in protein synthesis. before the operon before the operon are set of genes that code for amino acid. All right, for real, you know that proteins are from amino acid. All right, so now we say that the operon has a promoter segment that is divided into what? Activator and repressor. Right, this is a promoter segment here. Right, this is divided into activator and repressor. Although here we don't have a label, but yeah, here we have it. Okay, this is uh okay, the promoter segment is divided into activator and repressor. Okay, here is just the repressor shown, but definitely know that yeah, the activator exists too. Okay, okay, it has the promoter segment divided into activator and operator and repressor operator. Okay, I think that's well enough. So now next we see that in the absence of lactose, this is just like from the picture. Okay, I think you're just trying to talk how the repressor segment was. So that in the absence of lactose, the operant repressor binds the operator and transcription is blocked. Okay, now in the presence of lactose, the operon, it can be called lac operon or operon. Okay, the operant repressor is released from the operator and the transcription proceeds at a slow rate. Okay, so this is the operator repressor, right? So this is just like what we are talking about, the segments of the promoter. All right, the promoter can actually can either be repressor or promoter. Okay, now next we see that when the body needs proteins, activator molecules, which could be any substance, bind to the activator operator. Okay, when the body needs protein, the activator molecules bind to the activator. Now, when they bind to the activator, this stimulates RNA polymerase to begin work of transcription in the synthesis of what? Messenger RNA. Okay, when the body needs protein, the activator comes in place. Okay, now this stimulates the RNA polymerase to begin work of transcription in the synthesis of messenger RNA. Okay, so this is just like still showing us the operon. Okay, and here like this, all we have here is the promoter segment, and the promoter segment is divided into activator and inhibitor gene. Okay, let's proceed. Now we say that when the body needs proteins, the activator comes in place. Okay, now the binding of repressor substances to the repressor operator inhibits what? transcription, right? When the repressor substance come in, transcription is inhibited. Now, next we say that inducer molecules could also bind to the repressor operator to inhibit it. Okay, if the repressor is binding and inhibiting transcription, if inducing molecules bind to the repressor, it will stop the action of the repressor. Hence, all right, it will promote transcription. Now, next we say that regulation is predominantly a negative feedback mechanism. Okay, the regulation is dominantly a negative feedback mechanism. Do we need more proteins? Do we, or don't we need more proteins and stuff like that? All right, so this is basically a negative feedback mechanism, right? So guys, with that we have answered our question on an operon. Okay, operon segment, and see you guys in the next tutorial, and bye for now.